Today we will learn about photochrome. So let's begin. Plants have acquired a high degree of developmental plasticity to optimize their growth and reproduction in response to their ambient environment, such as light, temperature, humidity, and salinity. Plants utilize a wide range of sensory systems to perceive and transduce specific incoming environmental signals. Light is one of the key environmental system to perceive. Light is one of the key environmental signals that influence plant growth and development. In addition to being the primary energy source for plants, light also control multiple developmental processes in the plant life cycle, including seed germination, seedling dioteolation, leaf expansion, stem elongation, phototropism, stomata and chloroplast movement, shade avoidance, circadian rhythms, and flowering time. The light mediated changes in plant growth and development, independent of photosynthesis are and are called photomorphogenesis. Light acts as a signal to initiate and regulate photomorphogenesis. Photochrome is a light sensing system that regulates photomorphogenesis. It is a photomorphogenetic. Light acts as a signal to initiate and regulate photomorphogenesis. Photochrome is a light sensing system that regulates photomorphogenesis. It is photomorphogenetic chromoprotein that absorbs red and fair red light most strongly and causes photomorphogenesis. It is a blue pigment with a molecular mass of about 125 kilodalton. The photochrome pigment is found to be present in two photoreversible forms. One is red light absorbing PR form, that is 666 nanometer, and second is far red light absorbing PFR form, that is 730 nanometer. Let's know about the features of PR form. It absorbs at a peak of 666 nanometer, bright blue in color and in active form. It is the form synthesized in seedlings. When PS absorbs red light, it is converted to the PFR form. Now, let's know about the features of PFR form. It absorbs at a peak of 730 nanometer, green, that means olive in color and active form. Second point is the PFR form is the active form that initiate biological responses. Third point is when PFR absorb far red light, it is converted to the PR form. Next point is PFR can also spontaneously revert to the PR form in the dark over the time, that is dark reversion. Photochrome in all plants is synthesized in dark entirely as PR absorption of red light. PR converts it to PFR and absorption of far red light by PFR converts it to PR. However, the absorption spectra of the two forms overlap in the red region of the spectrum. When PR molecules are exposed to red light, most of them absorb it and it converted to PFR. Thus, small amount, thus the portion of photochrome in the PFR form after saturating irradiation by red light is only about 85%. Similarly, very far Similarly, very small amount of far red light also absorbed by PR makes it impossible to convert PFR entirely to PR by broad spectrum far red light. Instead, equilibrium of 97% PR and 3% PFR is achieved. This equilibrium is termed the photostationary state. The equilibrium is termed the photostationary state. Both PR and PFR form also absorb blue light. Hence, photochrome effects can also be mediated by blue light. Photochrome is a family of chromoproteins with a small covalently bound pigment molecule. Photochrome proteins occur as a dimer of two 125 kilodalton polypeptides, each with a covalently attached pigment molecule. The pigment is called the chromophore. It is a linear tetrapyrrole termed photochromobulin and similar in structure to mammalian bile pigments, that is bilirubin. Photochromobulin photochromobulin is synthesized in the plastids and its precursor is delta amino labulinic acid. Together, the apoprotein, that is polypeptide chain, and its chromophore make up the haloprotein. The polypeptide chain consists of an N-terminal photosensory domain and a C-terminal regulatory domain connected by a flexible hinge region. The N-terminal domain can be further subdivided into four subdomains: N-terminal extension, insert NTE, and per per arts and SIM, that is PAS and GAF and PHY, while the C terminal domain divided into two subdomains, the PAS related domain PRD containing two PAS repeats and the histidine kinase related domain, insert HKRD. The chromophore is attached to a conjured cysteine residue in the GAF domain. Assembly of the apoprotein with its chromophore is autocatalytic and occurs spontaneously. When the chromophore absorbs light, there is a slight change in its structure. This causes a change in the conformation of the protein portion to the form that initiates a response. The action spectrum of the light needed for photochrome mediated responses shows a peak in the red at about 666 nanometer. These responses can be reversed by an application of far red light peak at 730 nanometer soon. After the red treatment, the change in absorption is caused by the conversion of the photoreceptor from one structural form to another. The red absorbing form changes to the far red absorbing form when it absorbs red light, that is 666 nanometer, and back again when it absorbs far red light, that is 730 nanometer. Upon absorption of light, the PR chromophore undergoes a cis-trans isomerization in the C15 and C16, that is carbon 15 and carbon 16 double bond between the C and D rings of the linear tetrapyrrole. However, a recent NMR analysis showed that the A pyrrole ring around C carbon 4 and carbon 5 double bond rotates during photoconversion. There are two different classes of photochromes with distinct properties. These have been termed type 1 and type 2 photochromes. Photochrome proteins are encoded by photochrome gene family termed PHY. Its five members are PHYA, PHYB, PHYC, PHYD, and PHYE. Type 1 photochrome, that is light label, which is encoded by the PHYA gene, is abundant in etiolated tissue. Its concentration decreases rapidly upon exposure to light as a result of transcription inhibition, mRNA degradation, and proteolysis. Type 2 for Type 2 photochrome, that is light stable, which is encoded by the PHYB, PHYC, PHYD, and PHYE genes is present at low levels. Is present at low level in both light grown and dark ground plants. Because its genes are constitutively expressed at low levels and the protein is stable in the PFR form. Phytochrome responses can be distinguished by the amount of light required, the number of the protons impinging on unit surface area is termed as fluence. Its unit is mole per meter square. Fluence rate, that is fluence per unit time, is termed as irradiance. Its unit is mole per meter square per second. Different phytochromes responses occur at different light influences. These responses fall into three major categories based on the fluence required. Very low fluence response, that is VLFRS, low fluence responses, and high irradiance responses, that is HIRS. VLFR can be initiated by fluence as low as 0.0001 micromole per meter square and they saturate at about 0.05 micromole per meter square. For example, seeds of Arabidopsis can be induced to germinate with red light in the range of 0.001 to zero point 0.1 micromole per meter square. Because the amount of light which is needed to induce response is minute, so very small amount of the total phytochrome is converted to PFR. Thus, far red light cannot reverse VLFRS. LFRs are initiated by fluence of 1 micromole per meter square and they are saturated at 1000 micromole per meter square. The classical red and far red LFRs are initiated by fluences of 1 micromole per meter square and they are saturated at 1000 micromole per meter square. The classical red and far red photoreversible responses of photochromes which are discovered by Hendricks and Borthwick are examples of LFRs. These responses show photoreversibility because the far red light reverses the responses. Both VLFRs and LFRs can be induced by low fluence. The total fluence is a function of two factors, the fluence rate and the irradiation time. Thus, a brief pulse of red light will induce a response provided that the light is sufficiently bright, and conversely Very dim light will work if the irradiation time is long enough.